to take a look at some animations, 3 short ones which are going to review muscle contraction, particularly with the ATP and the calcium involvement, troponin, tropomyosin, those kind of things. These are kind of the things that we talked about last time. And let me find my numbers here right quick. Hang on just a minute. Take a look at a couple.
Nervous stimulation excites the skeletal muscle
They
cell.
look like little scared faces.
I'll get our lights off so we can see a little bit
An
better
action
here.
potential will trigger the release of calcium ions into the sarcoplasm. Calcium ions bind to the regulatory proteins and trigger contraction.
So quick overview, we're going to get the details of that a little bit later. Again, what were the 2 things needed to cause a muscle contraction or needed for muscle contraction?
Calcium
And
glycogen.
ATP, ATP and calcium. If those 2 are present, we get a muscle contraction, or at least we get force generation at our muscle contraction. All the way down to this one. Let's take a look at the contraction cycle. This is kind of where we were ending upon last time. We just talked about this briefly.
A muscle action potential triggers the release of calcium ions into the sarcoplasm, initiating a contraction cycle.
So they are coming from that sarcoplasmic reticulum into the sarcoplasm.
A single contraction cycle consists of 4 steps. First, ATP, attached to the myosin crossbridge, must be hydrolyzed to provide the energy needed to move the myosin protein. Second, actin and myosin filaments temporarily bind together. Third, the release of the phosphate molecule triggers the power stroke. The myosin filament rotates, releasing the ADP. The force of the rotation slides the actin filament toward the center of the sarcomere. Fourth, the myosin head and actin are detached when another ATP molecule binds to the crossbridge. This ends one contraction cycle.
Okay, we talked about that, and that was the importance of ATP in the contraction cycle. Let's talk more about calcium now, I think is our next one. And we'll go to this. I think this is calcium is the next one.
This cycle continues as long as ATP hydrolysis continues and nervous impulses continue to reach the cell.
Once the action potentials stop, then calcium concentration will drop and the contraction cycle ends.
So we have to have calcium to do this. And then we'll kind of, one last one here. And this is going to kind of link what happens at a molecular level at the sarcomere to transferring of actual force generation on the skeletal structure. So we go from kind of micro to macro on this one.
Contraction force is transmitted through a series of structures to produce movement.
By the way, there's an error here. See if you can catch what's going on. It's an
Movement on
error.
myosin filaments pulls actin filaments,
The error's going
sliding
on right
toward
now.
the center of the sarcomere. The actin filaments are connected to the sarcolemma through dystrophin proteins. The shortening sarcomere causes the sarcolemma to move.
Don't worry about dystrophins. I'm not going to ask you that one.
As the sarcolemma is shortened, the entire muscle cell contracts. The more muscle cells that contract, the more forceful the contraction. The force of the contracting muscle is transmitted through the connective tissues of the skeletal muscle to the periosteum of a bone. The contraction proceeds in a manner that brings the insertion toward the origin. In summary, the movement of an actin filament sliding along a myosin filament is transmitted through a series of structures to move bones and body parts. This movement results in the most movable attachment site of the muscle, the insertion, moving toward the least movable attachment site of the muscle, the origin.
So now we can see how we go from that micro level, molecular level at the myofibril all the way up to moving an entire skeletal part, entire body part.
You did say there was an error in the beginning,
There
right?
is an error, yes.
Is it the actual shortening of the...
Yes, they described it correctly. The oral description was correct. if we start this out again,
Contraction
what's
force
happening
is
here?
transmitted through a series of
showing the-
structures.
let me pause that for a minute. They're showing the shortening of the sarcomeres, but they're not showing the filaments sliding past one another. They're almost showing it like the actin and myosin are actually shortening themselves, and they don't shorten. They stay the same length. You just simply have the myosin and actin sliding along. Maybe that's a limitation of what they can do with the animation here, but nonetheless, it's
So
not correct.
it's just the space between them shortens.
Yes.
Like they're here and they
Well,
just slide together.
on- They just kind of, right, they just go slide right on past one
Sliding
another.
toward the center of
Right,
the
so
sarcomere.
they actually just showing the whole thing
The
shrinking
actin
as a unit.
filaments
That's
are
not what
connected to the
happens.
sarcolemma through
The
distal
sarcomere does
connections.
shorten, but the
The
myosin
shortening of
actin filaments do not
sarcomere
shorten.
causes the sarcolemma to
I'm trying to talk over that, sorry. Yeah, so the myosin actin, they slide over one another, but they don't shorten themselves.
Okay, what about the zetas part? Is that not shortened?
Not really, not really. They're just being drawn closer together. So that's just two ends. Think of two caps on a big cylinder. They're just actually being drawn together. So the Z-discs really don't change.
Have you ever thought of,
Sarcomere length does, but no other links really change.
you ever thought about contacting them and telling them what they did?
Oh, I can't remember. I think I may have contacted a lot. There's a bunch of errors on some of these. I contacted them.
Like the Hawk system, I made $30 off that.
A lot of times the animations are made by people different than the authors of the textbook, and it's sometimes difficult to get those fixed. All right, this is, I believe, where we left off last time, and we talked about the role of ATP, then the role of calcium in muscle contraction. And I think, did I show you this little thing last time? I don't think I did. Okay, so this just details the events at the sarcoplasmic reticulum. And on my conceptual model here, we've got two different proteins in that sarcoplasmic reticulum membrane. We've got these ones I've illustrated in green. These are always going. They're always pumping calcium to the inside. They never quit working. They're always pumping any free calcium to the inside. The other ones are what we call voltage-gated ones. That is, they could be open or closed. Under certain kind of triggers, they open up, and then they basically allow calcium to rapidly diffuse to the outside from high concentration to low concentration. So in this particular state, we're in resting state. The muscle's not contracting. It's not been signaled by the nervous system to contract. They're closed up, and we can build that up. So we'll put this into motion. We've got some calcium ions that are starting to be scavenged back in occasionally by those always ongoing calcium pumps. If we get a trigger to open up those blue proteins, that is the gated ones, if they open up, then what happens is they come out very rapidly. Now you also notice that some calcium is being flooded back or being brought back in as well. These pumps are still working, but they simply can't compensate for the calcium that's going out. Not until those gates get closed back again. So if we see that one more time, go ahead.
The blue one's taken out of?
Yes, yeah, and that's passive. They're just simply an open and closed gate. There's no ATP involved with them. If they're open, they're